Hello, class. So this is the third part and final part for the series of fatty acids and triacylglycerol metabolism. Recall in the first part of the series, we talked about consuming carbohydrates and how it ends up going through our bodies to eventually store triacylglycerol in our tissues. And then the second part of the series was that triacylglycerol that's in the tissue being going through degradation and then now being able to be mobilized and go into our body. But in order for it to go into the mitochondria to begin to be broken down for energy source, it needs to be activated by binding to the coenzyme A. And then from there, it gets attached to the carnitine to be able to transport inside of the mitochondria, and then it gets reattached to a new coenzyme A, and then goes through the beta oxidation and the breakdown of the fatty acid. And so that oxidation aspect of this process is the main focus for the part 3 of this talk. So once again, just bringing up this table as a reminder, you have this fatty acid that gets activated by being attached to the coenzyme A. Remember that this happens in a two-step process, which is an ATP dependent type of reaction where the fatty acid is attached to AMP. And then from there, on the second step, the fatty acids get transferred over to the coenzyme A. And from there, where the products will be the AMP and the pyrophosphate group. And also what we need is the activated fatty acid, which is the acyl coenzyme A. And so now is ready to begin the process of being transported into the mitochondria. But in order to do that, that fatty acid now needs to be attached to carnitine. And then once it is attached to that carnitine, it gets transported into the mitochondria. And then from there gets reattached to the coenzyme A that's within the mitochondria. And now we have the acyl coenzyme that's ready to go through the beta oxidation. So the stages of fatty acid oxidation is listed here. There's three major stages. One is having this fatty acid where it's being cleaved and broken down into acetylcoenzyme A, but as it's being cleaved, there's going to be electrons that's going to be released to these cofactors, which will transfer electrons to the respiratory system, where electron gets transferred to oxygen to produce metabolic water here. And also it produces ATP, which is our energy source. But also what creates some more water and ATP is that acetyl coenzyme A. It goes through the citric acid cycle, producing carbon dioxide and more electrons that goes into these cofactors that transfer electrons into the respiratory system and produce more water and more ATP. And so these are all the aspects of how ATP is being generated for that from initial breakdown of the fatty acids. So this is just an example of a fatty acid that they use, the palmitoyl, that is attached to the coenzyme A. What happens is you have an alpha carbon that is directly next to the carbonyl group, and then you have a beta carbon that's next to the alpha carbon. So in this um, in this oxidation step, a double bond is produced in between the alpha carbon and the beta carbon. Once again, this is the alpha carbon, is next to that carbonyl, and this is the beta carbon. Also notate that it has this trans stereo isomer configuration. This is going to be important for later. But when you go through this oxidation, it utilizes this enzyme, acyl coenzyme A dehydrogenase, where electron is transferred into the FAD cofactor to reduce it to FADH2. And then there is another protein called the electron transfer flavor protein that also gets electrons to be transferred over to this protein, and then it gets transferred to another protein that is a flavo protein, and it's an ETF um, in the acronym for it. But this one has ubiquinone, ubiquin, ubiquinone, excuse me, oxido reductase, and then from there, the electron is transferred into the respiratory system, and once again, electron goes to the oxygen, producing the metabolic water, and also producing ATP. Within this process, as far as ATP that's generated is 1.5 ATP. So this is kind of shown here in this slide about this process where you have your fatty acid and you have your alpha carbon and your beta carbon, and produces that double bond in between them, and electron gets transferred over to the FAD to produce the FADH2. But then you have this ETF that also gets transferred electron be transferred electrons onto it, producing this FADH2. And then there is a another flavor protein that has an iron sulfur protein that's in there. So that's why they just wrote in short iron sulfur, but in whole, this is a whole protein. And so from there, it actually gets reduced when the electron is transferred over to this new flavo protein. And then from there, once the iron is reduced, then the electron is transferred over to ubiquinone to produce ubiquinol, and then from there gets transferred into um, the respiratory chain to produce the ATP. And once again, it's 1.5 molecules of ATP is generated in this stage. So after we just covered this section, now we're going on to back to the fatty acid that just got oxidized to form that double bond between the alpha carbon and the beta carbon, and having this trans stereoisomer. Well, now in order for water to be added, this enzyme that produces an hydroxyl group on the beta carbon, in order for this to happen, this particular enzyme is stereospecific, and so it needs the transform in order for this reaction happen within this enzyme. So this enol enol coenzyme A hydratase produces the hydroxyl group on that beta carbon, and you're now starting to see this acetyl coenzyme form, but we need to have not a hydroxyl group here, but a carbonyl group there. And so now it goes through another oxidation to produce a carbonyl group, but this particular enzyme is beta hydroxyacyl coenzyme dehydrogenase. So just notice this the initial dehydrogenase has additional name that's attached to it because the beta is at that beta carbon, and then the hydroxy, it has a hydroxyl group. This one it's just a plain the beta carbon doesn't have any sort of functionality to it, and so that's why it doesn't have this extra name that is attached. But with this oxidation step, electron is transferred into NAD plus to produce NADH, which releases transfers electrons to NADH dehydrogenase, which transfers electrons also to the respiratory system, and um, electrons is transferred to oxygen, producing that metabolic water again and producing ATP as well. And for this step in particular, it generates 2.5 molecules of ATP, where with this step here, it produces 1.5 molecules of NAD plus. ATP, 1.5 molecules of ATP, excuse me. And so overall, within this stage right here, it produced 4 molecules in total of ATP per pass of oxidizing the palmitoyl coenzyme A. And so now we have this acetyl coenzyme that is almost there, but it's just attached to this carbonyl. And so what you have is the cleavage that happens, and the enzyme that is a part of that is the thiolase, and it attaches the coenzyme A to this carbonyl, where now we just have another fatty acid, but this 2 carbon short, and where that 2 carbons is now part of this coenzyme producing the acetyl group. And then from here, it goes through the process all over again, where it gets oxidized, produces the double bond between the alpha and the carbon, the alpha and the beta carbon, and then from there water is added, produce the hydroxyl group, and then oxidation happens again to produce the carbonyl group, and now it can be cleaved again to produce acetyl coenzyme. And so this loop happens over and over and over, and it goes in pairs of 2. So you end up in this stage where you only will have 2 carbons that is left. So it will naturally end with an acetyl coenzyme. And so once again, just looking at the steps with fatty acid degradation in comparison to the fatty acid synthesis that we spoke about earlier, just notice as you have that oxidation happen where the alpha and the beta carbon gets that double bond, and then you have a hydration that's happened. Remember, this is a stereo specific type of enzymatic activity, and produces that hydroxyl group and then gets oxidized again to form that carbonyl, and then you have your cleavage that happens right here to have that activated acetyl group. And so remember, it goes in the reverse. For the fatty acid synthesis, it has condensation happen with the activated malonyl group with the acyl group. You have that condensation that happens, and then we need to get rid of this carbonyl group. So it goes through a um, reduction to produce a hydroxyl group, and then dehydration to remove the hydroxyl group to form that double bond, and then reduction again to finally have the saturated fatty acid. So, as far as with the oxidation and just looking at the chemical equation that's involved with this, um, they used a particular example with this palmitoyl coenzyme A, and remember that this has 16 carbon lengths, and so in order for going through the process of oxidation, you only need to go through 7 passes because you're going to end up with an acetyl group at the end as a product. And so if it goes through seven passes, that means 7 acetyl coenzyme A is produced, and you have one remaining. So you end up having 8 in total as far as the acetyl coenzyme A. It goes through 7 passes, so it needs 7 coenzymes to be attaching as things are being cleaved. And it goes through 7 passes of transferring electrons over to FAD, and then also the NAD plus. And so thus also you're going to end up having 7 passes of water that's going to end up being um, um, going to be needed because of the hydration step. And so there, thus you have a product of 7 FADH2 that ends up going to produce ATP, and then also you have 7 NADH that also ends up going on to producing ATP as well, and then you have some protons that is also released. And so with that, that note of having the FADH2 and the NADH, remember that in each pass, 4 molecules of ATP is made in each pass. And so since it's going through 7 passes, it's 7 times 4, and you produce that 28 ATP, and also 7 metabolic water that is happening. So once again, we just talked about this stage 1 of having oxidation, hydration, oxidation again, and then a cleavage to make the acetyl coenzyme A, which will go through the citric acid cycle. We'll talk about that later. But the breakdown of the fatty acid itself generates electrons, and the citric acid generates electrons that go to these cofactors, that go to our respiratory system, and end up producing ATP and H metabolic water. And the product for the citric acid once again is carbon dioxide. So the major products for this oxidation is carbon dioxide, ATP, and water. So, as far as generating, how much ATP are we getting off of this one molecule of the palmitoyl coenzyme A? So what we did discuss was you'll see 10.5 plus 17.5 as 28 ATP. And so as far as the citric acid cycle, it produces more ATP, but we'll go in more in details about that later. And you have an overall of 108 ATP that is being created. But in order for the fatty acid to be activated, to be attached to this coenzyme A, it actually needed two molecules of the ATP. So remember, that was that activation stage. If we go back to this slide, talking about the fatty acids being attached to the coenzyme A, and it utilizes ATP. So it's two molecules of ATP that is being consumed. And so thus, for that complete oxidation of the molecule of palmitate, it yields 106 molecules of ATP, not 108. So don't get tricked by that, because two molecules of ATP is going to be taken away from the 108 that was produced to have an overall um, uh, uh, overall yield to be 106 molecules of ATP. So what we talked about before was the condition of having fatty acid that has no double bonds in its chain, so it's fully saturated. When we have unsaturated fatty acid, we're going to have to do something different, because something else happens where you end up having this cis configuration. And remember when I talked about the enol coenzyme A, it needed trans, and so now it's not going to work effectively. So how does our body overcome this? So with this fatty acid, you can do the normal cleavage, because there is no double bond here, and so three acyl coenzyme can be produced. But now we get stuck here where there is a double bond that is a cis configuration. So what happens is, before we get to that step of having an alpha and beta carbon that has a double bond with it, in order to get to that stage, you need the enol coenzyme A isomerase. So this particular enzyme goes through isomerization, and so it produces that double bond between that alpha carbon and that beta carbon, and now it can go through the rest of the cycle of the beta oxidation without any issues. Now there is another situation where you might have two cis double bonds. And so once again, it can go through the normal cleavage because there is no double bond that's blocking anything. And so you have three acyl coenzyme that's released. But now, once again, you have this alpha and beta carbon, and you have the double bond that is a cis that's attached to the beta position. So from there, it can go through the isomerase again, and have that double bond move to be in between the alpha and beta carbon. So we're good to go again. And now acetyl coenzyme A can be released. But then once again, it goes through a stage of producing a double bond in the first oxidation. And so that's where this new double bond is coming from. This is not the same double bond that was before. Remember, in the first stage, let's just in case you get confused. Remember, in the first stage, it's an oxidation step on a alpha and beta carbon, and produces that double bond. That is where this cis double bond is coming from. This other, not cis, trans double bond is coming from. Excuse me, but we still have this original cis double bond that is messing things up, and so now it needs um, a different enzyme to help fix this issue, where you use a reductase type of enzyme to reduce the double bond and now have only one single uh, double bond that is produced, that is in a trans position, but it's not at the alpha and beta carbon, and so it goes through the isomerase again to make sure that the alpha and the beta carbon has that double bond, and then goes through the regular oxidation of the the fatty acid. And so with our bodies, it has this self built in regulation. And so with one thing with when we eat our carbohydrates and have glucose that's in our body, whenever there is a low glucose, remember that glucagon is produced, and starts some phosphorylation of some proteins, and then from there, what happens is when you have insulin that is around, that actually releases the phosphoryl groups that is attached to these proteins, where now it can be activated to produce more malonyl coenzyme A, which you know is the building block to creating fatty acids. But when there is too much malonyl coenzyme A that's around, it starts to inhibit the fatty acids from starting to oxidize, because we know from there we don't have enough fatty acids that's being produced, because these needs to break down more. So that's why it's blocking that, the breakdown of fatty acids, because the body already thinking if it has too much malonyl coenzyme, that means not enough fatty acids are being produced. So we don't need to break down the fatty acids that we already have. So that is why it's blocking, and act as an inhibitor. And so what else does inhibits a process is producing too much NADH. So if your ratio of NADH to NAD plus is too high, that means we're breaking down too much. And so the enzyme that is a part of the beta oxidation is also inhibit, and so now is not breaking down fatty acids as much because it's too much of concentration of the NADH. So we're breaking down too much. And so one other thing is when there's too much acetyl coenzyme, that means that things are being broken down too much as well. And so it will block the thiolase. And so remember, the thiolase is the one, is the enzyme that is doing the cleavage. So just making sure you remember the thiolase. That's when we have that carbonyl. In the carbonyl, we have this acetyl coenzyme A that's right here, but the only thing missing is that cleavage. That's when you have too much acetyl coenzyme around, it blocks this enzyme and it inhibits it. And with that, that is the lecture on the beta oxidation of fatty acids, and we just ended it of how our body kind of regulates all this breakdown of fatty acids and build the synthesis of fatty acids.